Good afternoon, everybody. Welcome to our webinar this afternoon, which is vaccine focused and will cover current methods and approaches for viral clearance. Before we start the webinar, just some little bits of housekeeping. If you have any technical issues, please send a short message in the questions box on the right-hand side of your screen. During the webinar, RSSL encourages you and invites you to ask questions. Please use the questions box on the right-hand side of the screen. At the end of the webinar, time permitting, we will try to answer as many of your questions as possible. If we can't get around to everybody's questions, we'll answer those offline and share them with you alongside the slide deck, which you will be presented with today. Excuse me. Just a little bit about myself. So I'm Gaynor Johnston, and I'm the biopharmaceutical lead at RSSL. I joined RSSL on the first day of lockdown, which was a very interesting time, as we can probably all remember. I work with an array of clients from large pharma all the way to virtual companies. My clients might be early phase looking for biomolecular services to support characterization and release. They might be calling on our specialist cell potency and mass spectrometry capabilities, or they could be manufacturing for trials or commercialization, and they might need our microscopy team, for example, to support them with unidentified particles in their biotherapeutics. No 2 days are ever the same at RSSL. Before we move into today's webinar, I'd just like to make you aware of our other webinars in our series this year, the ones that are coming up, as you can see on the screen. Please head over to our website if you'd like to join those, and you'll also find Tim's monthly regulatory updates as well on our webinar. Who are we? Let me just give you a little bit of the story of RSSL, if I may. So we're a growing CRO. We opened our 4th lab space, which is a sizable 26,000 square feet at the end of 2023. RSSL have been around for over 30 years and are based exclusively in the Reading area. If you don't know where Reading is, we're about 40 minutes west of London and close to Heathrow. Our 4 sites host over 350 scientists, and we have 12 different technical areas that we specialize in analytically. We have clients globally in Europe, the US, and as far away as Singapore and Australia. Being a relatively compact CRO, this gives us a cross-site agility to manage complex multidisciplinary projects. This is supported with around £20 million worth of equipment in our Reading-based facilities. We're very fortunate to have a multi-billion-dollar parent company, food snacking giant Mondelez, who own a multitude of brands that you've probably heard of, like Cadbury's, Oreos, and Philadelphia, to name just a few. This gives us access to funding to support expansion as our markets grow and diverge. One slide, it's quite a busy slide, so I'm not going to go into all the detail of that, but as you can see here, from our specialist labs, we offer a diverse range of biological, microbial, chemical, and physical analytical services across all phases, from development through to commercial release. We are very well placed to support all of the new Annex 1 regulations and have a highly knowledgeable technical team who can help advise and help throughout your manufacturing. As you will see from this slide, for vaccine manufacturers, we also provide biomolecular analytics from our biosafety level 2 facility, and we have capabilities for protein therapeutics, LNPs, and viral vectors. This can be for characterization and release testing purposes, or also on a non-GMP information-only basis. Viral clearance is also available with methods like those on screen. We also do a lot of primary packaging testing, including vial and stopper testing. And as you can imagine, we were extremely busy with this during COVID, but we also test other components typically used in novel biotherapeutics, like plasmid bags, connectors, tubing, those kind of items. We can support you with your E&L studies and can generate a contaminant library bespoke to your product and process to quickly identify foreign bodies in your biotherapeutic. ICH Q1A stability studies are also offered, including photostability and force degradation studies from our dedicated on-site stability rooms. For further details on any of these services, please visit the web link below or contact me directly. I will be most delighted to hear from you. Please allow me today to introduce our guest speaker, Dr. Tim Sandle. I'm sure some of you have heard of Tim and subscribed to his publications. Tim has over 25 years experience of microbiological research and biopharmaceutical processing. He's a member of several editorial boards and has written well over 600 book chapters, peer-reviewed papers, and technical articles relating to microbiology. Tim works for a pharmaceutical manufacturer in the UK and also finds time to be a visiting tutor at both the University of Manchester and UCL. Tim is a busy man. Let me now hand over to Dr. Tim Sandle. Thank you.
Okay, thank you very much. Just do a screen swap. Brilliant. Okay, well, welcome everybody. Welcome to this webinar. This is a slightly different subject to some of the earlier ones. So what we're going to be focusing on today is with different approaches and current methods and risk framework for viral clearance. So the areas that we're going to be having a look at, we're briefly, just for those who are unfamiliar with sort of viruses and viral safety in general, we'll look at viruses and vaccines, then we'll look at current regulatory guidance and some of the risk factors, and then we'll move into sort of more of the detail around viral inactivation and virus removal, and then bring all these disparate elements together in a sort of more synergistic approach. And then we'll have a look at how we can create dedicated viral secure areas, and also the likelihood or otherwise of viral transmission through the air within controlled environments and clean rooms, and some of the mitigations that we can put in place for that. But let's begin with viruses. So we've got a colourful slide with viruses of all different geometric shapes. So viruses are found in almost every single ecosystem on Earth, and they are the numerically highest biological entity. And there's obviously a long-running school of thought about whether or not you can classify viruses as a form of life, but for these purposes, we're talking about them as being biological entities. So given the ubiquity of viruses, they pose a challenge to many aspects of biopharmaceutical manufacturing. And these challenges not only relate to viruses per se, or the viruses that might pose specific problems to production processes, but also from the varied nature of viruses themselves. So we have the actual genetic material, the molecules of DNA or RNA that encode the structure of proteins upon which viruses act. We have the nature of the viral protein coat, or the capsid that surrounds or protects the genetic material. We also have challenges from the range and complexity of viruses and viral particle sizes. So we have everything going from sort of a simple helical and isohedral forms to more complex structures. And the more geometrically sophisticated some of these structures are, then the greater the degree that they can affect certain viral removal processes like nanofiltration. There are also risks presented from both viruses of concern in starting materials, and we also have risks presented from viruses as adventitious agents during bioprocessing. We need both a control strategy, a removal inactivation strategy, and a testing strategy. And we need to bring all these elements together. So viral contamination is a potential safety threat that's common to a range of animal and human-derived biologics. And it's continuously challenging because of the emergence of new viruses, the greater sophistication and range of different products, including viral vaccines. So ensuring biological safety is a challenging exercise. And there are some products that are of particular concern, and they're listed on the slide. So we have gene therapies, monoclonal antibodies, recombinant proteins, a whole range of blood and plasma products, animal, human tissues and fluid products, medical devices, therapeutic vectors, and of course, vaccines. And contamination of the production system can occur, and we need to recognise the different points where such contamination can arise, and then have well-developed viral clearance processes in place, and the activities of virus removal are complex, and they also need regular assessment. And we have a further challenge around the kind of concept of viral secure areas as well, and how we control materials and personnel and configure things like air filtration. Another thing is when we're discussing the sort of concept of viral safety, we can define this as trying to exclude or verifying that we can exclude viruses of concern, but the absolute freedom of extraneous viral agents is not easy to achieve, and some virologists would argue that it's an impossible task, not least because it's something that can't be verified due to the difficulties or impossibility of testing for every known virus. But what we can do is demonstrate that viruses and virion activity has been appropriately excluded for those viruses that we are particularly concerned about. So we can talk in the concept of excluding or clearing those viral pathogens of concern by viral inactivation methods or viral removal methods from our product streams. So we will have a set of different products and manufacturing stages that will present different points of risk. So we're going to have products that are made from in vitro culture of cell lines of either animal or human origin, products produced from in vivo cultures of cell lines, products produced from organs or tissues of human origin, and products produced from blood, plasma, and other human fluids. So they're all going to be particular areas of risk. Viruses are also important in vaccine development, and a lot of the acceleration of technologies around the time of the COVID pandemic has brought a number of these to the fore and has arguably helped to accelerate this area of science. So vaccines, including viral vaccines, obviously have a long and crucial role in human history and human medicine. And so they continue to improve lives through prevention, control and eradication of infectious diseases. And with viral-based vaccines in particular, then these rely on the antigenic properties of a virus or virus-like particle to trigger an immune response against an incipient viral infection. And because of the risks associated with live and inactivated viruses, then we need to have strict controls, and these controls include avoiding opportunities for adventitious contamination agents to affect our production processes. And we also need to ensure that the viruses that we are using ourselves are indeed appropriate. So where we're making virus vectored vaccines, these are laboratory generated viruses that are based upon replicating viruses, or they are based on non-replicating virus vectors into which we've spliced genes expressing antigenic proteins for our target pathogen. So a live vectored virus vaccine is something that's biologically active, and it produces virus progeny in the vaccinated host, but equally it may be attenuated for pathogenicity, and that can either be due to mutations in the vector because of the chimeric nature of the vaccine itself, or because the vector is used in a heterologous host, or it could be due to a combination of these factors. Whereas with a non-replicating virus vectored vaccine, this is a viral material that's so heavily, so severely attenuated that it cannot undergo a complete replication cycle once it enters into the infected host cells. We also have recombinant vaccines. So recombination is a description of a process in which nucleic acid sequences from two different parental viruses, in this case, are exchanged so that the resultant product, the progeny, contains sequences derived from both parents. And both RNA and DNA viruses can be subject to recombination. And there are different mechanisms through which we can achieve recombination. And these mechanisms are influenced or dictated, if you like, by the structures of the viral genomes. So for DNA viruses, we would attempt recombination by the physical breakage and rejoining of parental DNA molecules. And we could do this through different regions of sequence homology. For RNA viruses containing segmented genomes, then we can achieve gene exchange through the reassortment of individual parental genome sequences into progeny viruses, or we can do a combination thereof. So although we can still achieve this with recombinant products, we still have risks of viral contamination of these products. In terms of viruses of concern, the contaminated cell type, the contaminating virus, and the likely source of contamination vary. Some of the most common contaminants, particularly of our sort of starting cell cultures, include viruses like herpes virus, human adenovirus, para-influenza virus, and reovirus. But in addition, depending upon the source of our starting cells, we may see viruses like the virus of mice, or MVM, of which there are different infective species. So we have viruses like rodent protoparvovirus, and these viruses are common infectious agents of laboratory mice. Therefore, they also become viruses of cell cultures or cell lines that have a rodent origin. Another one, and because so many cell cultures use starting lines like Chinese hamster, ovary cells, which is a common basis for many biological drugs, then these particular rodent viruses can be viruses of concern. We also have vaccinovirus, which is a large complex enveloped virus, and also rhabdovirus as well, which is one of the perhaps more sort of novel emergent viruses of note, and this is a virus family where there's an association with rabies and also conditions like vesicular stomatitis in cattle and in horses. So we've got this whole range of different viruses. Some are going to be more common, others are going to be more starting culture or cell line specific. And we also looked at different types of viruses and different sort of shapes of viruses. But we also have another distinction that has a big bearing when we move into look at viral clearance, and this is whether viruses are enveloped or non-enveloped. So viruses can be enclosed in an envelope that's made of proteins, carbohydrates, and lipids. And an example of an enveloped virus is the herpes virus. But equally, viruses can be non-enveloped as well. And an example there is with parvovirus. Generally, but not exclusively, non-enveloped viruses are more difficult to remove in viral clearance studies. But obviously there are some nuances that affect that, and we'll look at some of those shortly. Okay, in terms of guidances, then one of the most useful documents that we can refer to is ICH Q5A, and a draft version of the latest iteration of this document has been in circulation for around 2 years and has gone through the process of receiving public comments. That process has closed, and the new version is going to be launched in a couple of weeks' time. And the European Medicines Agency has announced that it will formally adopt revision 2 of Q5A on the 14th of June, and it's expected that the US FDA will adopt the guidance very shortly afterwards. So the revision, there isn't time to go into a lot of detail, but it has been updated to capture sort of more detail about the scientific developments, the advances in biotechnology, particularly with the various iterations with viral vaccines that have taken place. It considers also a sort of wider expression of risk for ensuring that we can begin our pharmaceutical manufacturing processes without the presence of viruses. So it kind of signals to us that we should be putting in place and developing a viral strategy. So with a viral strategy, that's essentially 3 primary strands. The first is with preventing the entry of contamination into production processes by ensuring the appropriate quality of raw materials. The second component is with detecting contaminants by the characterization of cell banks and virus seed stocks, and also by putting in place a sound testing regime and testing various junctures of the manufacturing process of intermediate stages to verify that we haven't had adventitious entry of viruses. And then there are the third part, which is about the core part of viral clearance, which is incorporating different viral inactivation and viral removal steps into the vaccine purification process, so that we can be sure of removing any additional viruses of concern. So with sort of the core subject of virus clearance, we have a combination of viral inactivation methods and viral removal methods, and we need to ensure that we are using a combination of these methods, and most risk-based guidelines and risk assessment outcomes point to the use of at least 2 such methods, depending upon the level of concern and the patient population. Often that will extend to 3 measures. So if we have a look at viral inactivation methods first, then some common and well-practiced examples of viral inactivation include solvent detergent inactivation, and this is a relatively robust and well-established viral clearance method, and we'll talk a little bit about that in a minute. Low pH inactivation, which is particularly effective against enveloped viruses, and this creates pH conditions within which viruses lose the ability to replicate. So critical process parameters that would be important to introduce at this stage to track the robustness of the manufacturing step would include pH naturally, time, and temperature. And then there are other methods of viral inactivation, including heat, a sort of form of pasteurization. It is possible to inactivate viruses with microwaves, irradiation, or high energy light, although those latter ones are less commonly undertaken. So heat is the primary method that's deployed. So all 3 of those methods are generally more effective against enveloped viruses, but they have relatively limited effect against non-enveloped viruses. So we need to look at other methods like chromatography and virus filtration to deal with non-enveloped viruses. But let's look at some of these technologies in a little more detail. So moving to the solvent detergent activation. So this involves incubating intermediate product, which is most likely some form of protein solution, in the presence of an organic solvent. So that be a solvent like triene-butyl phosphate, and also with a detergent. So for example, something like Triton X100 or an alternative. And there's been a little bit of a push in recent years to sort of move away from Triton X, because it is quite a nasty substance. After the solvent and detergent process, we need to remove both from our product line once we have completed the viral inactivation process. And one way to do that is to use a chromatographic mechanism to ensure that we're extracting the pure proteins and removing the traces of the solvent or the detergent. And often we need the use of an additional chemical that will be acting as a sorbent, so a form of polymer would be quite typical. In terms of other viral inactivation methods, we mentioned some of them. So we have the potential for ultraviolet light, which needs to be in the UVC range. So it's the application of a low dose of radiation. So a typical virucidal wavelength would be 254 nanometers, and again that has a greater effect against non-enveloped viruses. Or we have heat, which is probably the most common method, although it's obviously going to depend upon the stability of the product that we're processing. So generally, and obviously there are different factors that can influence this, but in general terms, a temperature of around 70 degrees Celsius for 30 seconds will be able to inactivate most of the viruses that we're likely to experience as contaminants within our processes. We also have chromatographic methods, so certain affinity chromatography steps in pharmaceutical processing, such as use of protein A, can be optimized for virus clearance. So in particular, different forms of anionic chromatography mechanisms have been shown to be effective for the removal of viruses as we move a process downstream and we attempt purification. The degree to which this is effective is dependent upon the resin and the binding mode. We also need to have good control of the flow rate, use the right types of buffers, maintain appropriate temperatures, and also use the correct wash volumes. And all of these are factors that can affect the viral removal capacity. Perhaps a more effective method is membrane chromatography, and this is shifting to become more of the method of choice in large-scale biopharmaceutical manufacturing processes. So it's a preferred choice because it not only helps remove viruses, but it's also very effective at removing a range of other impurities. So in terms of virological control, the process works by removing viruses by an absorptive material like an ion exchange membrane that has ligand virus binding properties and carries a positive surface charge. Another advantage with this technology is that it has a relatively higher flow rate beyond the one we can achieve with conventional chromatography. But we also need to be careful and protective of our product yield, because one of the ironies is that the more efficient the process becomes at removing viruses, this can affect the protein concentration and also could have an adverse impact on other product characteristics like the hydrophobicity of the product. So it's something we need to be mindful of. Another way of removing viruses is nanofiltration, and here viruses are removed from a product by size exclusion. So almost all forms of nanofiltration are effective against large viruses, and large virus is something that would be defined as 50 nanometers or greater. And any kind of filter that gets classed by a reputable company as a nanofilter will be able to remove these types of viruses. For the removal of smaller viruses, so we're going 25 nanometers as a typical size, this requires slightly finer, slightly higher specification nano filters, and it also requires careful control of filtration parameters like time, temperature, and pressure. But if we get this optimized, then we should be able to achieve a logarithmic reduction of at least 4 logs of small viruses of both enveloped and non-enveloped types. One of the areas where things can go wrong if we don't adequately control those key parameters, particularly flow and pressure, is we could get aggregation forming, and that's something we need to seek to avoid. So the control of that is really important. It's also important for each one of these sort of individual approaches, whether we use them singular or in combination of viral inactivation and viral removal, that we validate the process so that we have a stronger feeling that we've got good patient protection. So it's important that viral clearance is demonstrated through qualification studies, and with such studies we're seeking to evaluate the ability of the manufacturing process to clear identified viral contaminants, and by clear that's the inactivation or removal. In addition, we need studies to assess the robustness of our manufacturing processes against potential adventitious contamination by viruses, and also to understand what's the capacity of our different steps in terms of the amount of product we can put through, and the conditions, the run rate that we can subject equipment to, because we want to avoid, as I said, particularly with nanofiltration, blockage to viral filters. They can easily become blocked by viral aggregates or through the denaturation of the proteins that are passing through. And then with areas like chromatography, is how long does our column media work for? At what point do we need to change that? So a thorough understanding and assessment of the technology that we're using in our manufacturing processes is really important to our qualification study, and capturing those parameters and putting them into routine use. Another important choice is the choice of viruses that we're going to use in the viral validation studies, and this is going to depend to a degree on the type of product, process, and also what the regulatory perspective is, and also the level of viral clearance that we want to achieve, as we might express in terms of logarithmic reduction. Good practice would dictate in most circumstances that we need to demonstrate that our process can remove both enveloped and non-enveloped viruses, and also particularly where we're using nanofiltration, that we can effectively demonstrate we can remove viruses of different shapes. So parvovirus, which we've already looked at, is isohedral in morphology, whereas something like the human immunodeficiency virus is spherical. Also, as I mentioned, that adopting one approach is rarely sufficient. And in most cases, regulators will push for 2 or sometimes 3 approaches. So we need to create a synergy of viral inactivation and viral removal methods. So you'd see things like manufacturers practicing membrane chromatography, filtration, and solvent detergent steps, or they might put in place a combination of filtration, solvent detergent, and heat inactivation, for example. It's also important, and this is this kind of concept of safeguarding the area in which we're producing products in, is that we also develop viral secure areas. So, and this is important to try and minimize the prospect of adventitious contamination, cross-contamination, particularly after we have put in place our key viral removal steps. So often we would be creating viral secure areas, and this concept becomes increasingly important the further we move downstream, the fewer process steps we've got to follow. It's also important in multi-product facilities as well, because that's a prime opportunity for cross-contamination. So the concept of a viral secure area would be to receive products that have gone through their viral removal and inactivation steps, and we are seeking to avoid recontamination from other products or materials or from people. So we'll have control spaces with separate air handling systems, and personnel entering those areas would be wearing separate clean room gowns, and we would have control of materials, and materials only being allowed in once, and having appropriate disinfection transfer protocols in place, and also ensuring that our viral secure area is protected by airlocks. We also want to control the ingress of viruses from the air, and here we'd have concern about viruses and viral genetic material. So to a degree, a sort of standard clean room heating, ventilation and air conditioning HVAC system will function to minimize the spread of airborne viruses through the standard ways that a clean room will work. So in terms of dilution, via air change rates, and by removal. And we can also inbuild further airborne protective measures, such as controlling the direction of the airflow and overall air patterns. And we can minimize the release of viruses from personnel by ensuring that people are wearing appropriate clean room gowns, but in particular wearing goggles and masks, because we want to reduce the ability of people to produce droplet nuclei through sneezing and coughing and so on. So to a degree, HEPA filters, which are much more orientated towards the control of bacteria and fungi, will also have a degree of ability to trap viruses or viral carrying particles. So HEPA filters are fairly well established. They're microfiber borosilicate glass. It comes in in the form of like a very stiff kind of paper that's folded into a concertina effect. And conventional HEPA filter will be able to capture 99.97% of all particles of 0.3 microns and greater. And research, going back to like the mid-1970s, shows that HEPA filters can capture a high proportion of airborne viruses, although the effectiveness of that does depend upon the number of viral particles and their relative sizes. And there are different test methods that can give us an idea of what could potentially be released through a HEPA filter system. So we can collect 4 viruses using special aerosol samplers and electrostatic precipitators, and then undertake plaque assays or carry out PCR for viral nucleic acids. But we can also enhance the HEPA filter through ionization as well. So again, the ability of ionization to inactivate viruses is something that's been established since the 1950s. And one of the things that was, again, pushed more through the sort of COVID-19 pandemic, but was to sort of retrofit HEPA filters with multifunction ion air cleaning systems. So a combination then of an ionization process and then the standard electrostatic attraction that's going on inside the HEPA filter can improve the ability to cleanse the air of incoming viruses. So this kind of technology is relatively energy intensive, but it can be at the same time quite effective of basically getting clusters of ions to surround airborne particles, including viruses, and essentially it's forming hydroxyls around each of the airborne particles and almost like punching a hole through the particle, which naturally inactivates the virus. Any part of a viral strategy and to support viral clearance is the testing of viruses. And there are different ways to test for these specific pathogens. Remember, something I said at the beginning is that complete viral clearance is not possible to prove, but we can screen for those pathogens of concern. So for that regard, PCR assays, polymerase chain reaction, is a good method of screening for specific viruses, so we can detect DNA viruses like herpes virus or RNA viruses like hepatitis viruses, which are encapsulated by protein codes, and therefore PCR becomes a suitable method for their screening. And PCR generally stands up as superior to other more traditional approaches like infectivity assays or even things like electron microscopy. An alternative is to PCR is next generation sequencing. So on one hand, real-time PCR is arguably easier to use, and some researchers say it's more tolerant of variable DNA quality. However, it's limited in its ability to screen for multiple targets. So next generation sequencing allows simultaneous analysis of different genomic loci, so we can get a broader screening for an array of different viruses. And some variants of next generation sequencing like shotgun metagenomics can allow us also to determine the order of nucleotides, so we can get far more specificity. It's also really important that we kind of draw all these different things that we've covered together in the form of a risk assessment, and there are obviously different risk assessment tools, and you look at ICH Q9, there's an array in there. Probably for this kind of control, I think that an approach like HACCP is a good one to use. So that's hazard analysis, critical control points, because it allows us to map out our process and to look for those points in the process where we might either have concerns about viruses remaining, viruses entering the process, and therefore where we need viral removal, viral inactivation, and also to consider points for testing. So the HACCP will allow us to sort of look at things like cell lines, other raw materials, certain intermediate process steps, and around finished product testing. And we also need to periodically reassess what we're doing. We need to consider how effective our viral inactivation and viral removal processes are. And one way we can do this is understanding sort of the viral titer that we start with and what we end up with at the end, as well as looking for specific viruses of concern. And again, this is all about building in this sort of approach into the validation, which we can strengthen and also develop as small-scale laboratory studies by running this kind of series of different viruses through our process. We also can gain comfort and verification by appropriately assessing the different parameters as we undertake our processes. So changes to critical process parameters might signal to us that the safety profile has altered, and that's particularly because we can't always, as with any approach to pharmaceutical risk assessment, put too much emphasis upon testing. Yes, testing is reasonably reliable, but it's not going to pick up everything. So all these various controls, particularly through filtration, solvent, detergent, heat, and so on, are really important. So just to conclude, we do have a number of viral risks with a range of biological products. We've looked at is the implication of these risks and where they might occur. We've looked at the importance of process controls, the importance of targeting controls through risk assessment, the importance of control through the twin processes of virus removal and virus inactivation. And we've also considered some of the testing techniques and the importance of overall viral security. So that brings the main part of the webinar to an end, and we can move into any questions.
Tim, that was excellent. Thanks for a very comprehensive presentation on viral clearance. We have had quite a few questions coming in, but we are a little bit low on time. So I'm going to quickly just ask a couple, if I might, Tim. Somebody has asked, what type of log reductions do you see from chromatography?
Again, I'd say a minimum of 4, but you can. I have seen up to between 5 and 6 before.
Another question we've had is, do viral clearance studies have to be conducted in process areas? No,
it is perfectly acceptable and possible to carry these out on a smaller scale in a laboratory setting, as long as the scale up has all of the sort of same controls. You've got the same robustness, the same control over temperatures, over flow rates, filtration, and so on.
So obviously it's easier to do it in a process area rather than taking it into a separate area. That's probably more common, I expect, is it?
Yes, it is. Yes, definitely.
Okay, one last question, Tim, I think, and then we're going to have to call time. And anyone that's emailed in, put a question on the chat box, we'll respond to you directly. Last question: which type of viruses give more cause for concern, enveloped or non-enveloped types?
It's a tricky one. I mean, it depends again on the particular process and pathogens of concern, but generally non-enveloped viruses are harder to deal with. They have that additional resistance because they don't have that immediate kind of target to work on, like the enveloped viruses. You've got something to hit immediately. Non-enveloped are a little bit more challenging.
Would you use the same clearance, methods to treat virus that were non-enveloped? So that's a part 2 question.
Yeah, I think it's to a degree, it's trying to remove, first of all, so chromatography, nanofiltration, and then probably heat is the optimal method to attempt.
Last resort. Okay, well, we're going to call time on that now, Tim. I've just got one more slide to show everyone. It'd be remiss of me not to mention we've got an open day coming up on the 26th of June. RSSL obviously has its brand new facility located on University of Reading campus. You're all very welcome to join us. There's an event registration link that you will find in your slide deck when you receive it. It's also on our website. Everyone is welcome to join. You do need to sign up for it because we are limited on numbers. Thank you all for attending our webinar. It's been great to have you along. I hope you've enjoyed it and you've had some learnings from today. We've appreciated the questions that you've asked. And as I say, anything that we couldn't respond to just now, we'll answer after the event by emailing you directly. If you want to catch up on any of our previous webinars or catch Tim's blog or regulatory update, please go to our website or keep an eye on our social media posts. Thank you all for attending today and enjoy the rest of your day. Goodbye, everybody.